टेस्टिंग कर ले। हेलो। हेलो। मैं क्या, माधवी बोल नहीं रही ना, माइक ऑन कर रही है, उसको बोलना एक बार बोल ले, टेस्ट करना है। दूसरा गेस्ट तो ज्वाइन नहीं हुआ, आपको ज्वाइन करा।
आप सुनो, उधर से आ रही है आवाज?
हेलो।
फिर मेरी वॉइस आ रही है उधर?
हाँ, वॉइस आ रही है। हेलो। हेलो। हेलो।
The person you have called is speaking to someone else.
मैं क्या, तो म्यूट करके क्यों बातें कर रही है, किससे कर रही है?
ईशा दीदी है, ईशा दीदी कह रहे हो आवाज आ रही है उधर?
तो मेरे से बात करना, जब मैं तेरे से कोऑर्डिनेट कर रहा हूँ तो उसको बीच में क्यों डाले जा रही है?
का कॉल आया है मुझे
तो
तो।
फोन काट उसका, मेरी बात सुन।
तो मैं आपको इधर से अनम्यूट करती हूँ, आप देखो आवाज आ रही है? सो गुड इवनिंग एवरीवन, दिस साइड माधवी।
बस ठीक है। फोटो भी ठीक है। एक थोड़ी सी लाइटिंग ऑन कर ले। फेस पे लाइटिंग नहीं है, डल है बिल्कुल। अगर रूम में है तो लाइट ऑन कर ले। 5:30 है ना? मैंने
हाँ
स्टार्ट
जी।
करके लाइव लूव कर दिया। मैं लिंक दे रहा हूँ, ग्रुप में डाल रहा हूँ। तेरे को दे रहा हूँ, ठीक है। मैंने सारा कुछ सेटअप कर दिया, बाकी चलाते रहना खुद ही।
ठीक है।
मैं तेरे को एडमिन, मेरी बात सुन, मैं तेरे को एडमिन एडमिन होस्ट ही कर देता हूँ, क्योंकि मैंने थोड़ा
ठीक है।
जाना है। तेरे पास वाईफाई है
ठीक।
ना?
हाँ जी, हाँ जी।
ठीक है, मैं तेरे को होस्ट बना रहा हूँ। जब गेस्ट आया खुद ही एडमिन बना देना।
ठीक है, ओके।
मैं तेरे को होस्ट बना दिया है। लाइव मैंने कर दी है, लिंक मैंने बना दिया है।
ओके।
राधे राधे।
Greetings to all. Any idea when the workshop will start? Serene ma'am?
Good evening, ma'am. We will be starting in next 5 to 10 minutes, ma'am. Like by 5:30, we will start the session.
Okay, thank you.
Okay, so good evening, everyone. This side Madhvi, your host for 3 days International Faculty Development Program on strengthening research and innovation ecosystem in higher education. So, which is jointly organized by Rayat Bari University, Mohali, Just Agriculture Education Group, ISA STR Society, Noida. So now I would like to invite our esteemed guest, Dr. Swati Verma, ma'am, Assistant Professor, Dr. YSP Parmar, University of Horticulture and Forestry, Noni Solon, to deliver their lecture. So before we start, I would like to introduce ma'am with you all. Ma'am is currently working as assistant professor at Dr. YSP Parmar, University of Horticulture and Forestry, Noni Solon. She obtained her PhD from IIT Roorkee in 2015. She did her MSc in biotechnology from Dr. Y.S. Parman, University of Horticulture and Forestry, known as Solin only, and BSc from CSK Agriculture University, Palampur. And during her doctoral program, she has worked on genetic engineering of cluster bean. She was awarded as National Postdoctoral Fellowship by SERB DST Government of India during 2017 to 2019. During her research tenure, she had been published several research papers in esteemed international journals. Many of them are on horticulture biotechnology research. So I welcome you, ma'am, on the behalf of everyone here.
Hello, I'm audible. Am I audible?
Yes, ma'am, you're audible.
Okay. So, good evening, everyone. Thank you, Madhvi, for this nice introduction. And I think the organizers of this program for giving me this opportunity to speak here. I can share the slides. So, are my slides
Yes,
visible?
ma'am, it is visible.
Okay, so on this International Faculty Development Program on strengthening research and innovation ecosystem in higher education, I'll be talking today about a research innovation which we can use to, isolate single cells, and thereafter we can go on for studying various biochemical, transcriptomic, and proteomic parameters in specific cells or tissues in plants. So I'll be talking about mining of microRNAs in apple buds and the potential regulators of dormancy release in apple. So I'm working as an assistant professor in Department of Biotechnology. We are a new college of Dr. Y.S. Parmar, University of Horticulture and Forestry, Solon. We are 6 to 7 years old only. And let us first of all understand what basically microRNAs are and why they are called microRNAs. So microRNAs, they are small 21 to 23 nucleotides, very small stretch. Normally a messenger RNA, it is greater than 1 kilobase pair, which is being translated into a protein. But these are single-stranded molecules, and these are non-coding, just a minute, non-coding microRNAs, non-coding molecules, which regulate gene expression by messenger RNA cleavage or translational repression. That is, they basically work in post-transcriptional regulation of gene expression. They either cleave a messenger RNA and thus they inhibit its translation into a protein, or either they inhibit translation, that is, they inhibit the messenger RNA from being converted into a protein. So you must be able to understand that why they are basically called post-transcriptional regulators of gene expression, because they are doing their work at the post-transcriptional level, not at the gene level. So there are post-transcriptional regulators of gene expressions, and most of the studies till date, they have been carried out on microRNAs in model plants like Arabidopsis, and very limited studies on microRNAs are available in woody tree species. So yes, So let us have a look how they are biosynthesized, how they are bio-generated within the cells, and what are their functions. So basically microRNAs, they are being transcribed from a gene which is microRNA gene. This microRNA gene, this is transcribed into a primary microRNA molecule, which is an imperfect hairpin loop containing molecule. Now, these are being processed by a protein complex which contains hyponestic leaves, that is HYL-HIL1, and dicer-like 1, DCL1 protein. This complex, it further processes these primary microRNA molecules into mature microRNA molecules, which are basically the 21 to 23 nucleotide stretch of small RNA molecules. These complexes, they are then transported out of the nucleus, and here they are assisted by RNA-induced silencing complex, which is a group of proteins. And this protein complex, it triggers the post-transcriptional regulation of one or several target genes through a base pairing mechanisms. That is, once they are outside the nucleus, they go on to repress their targets, which can be various messenger RNA molecules. Either they cleave them down by complementary base pairing at a specific site on the messenger RNA, and they are guided by this complex, RNA-induced silencing complex. This complex induces the breaking down of that molecule, messenger RNA molecule, and either they cleave the messenger RNA or they carry out the translational repression. That is, they inhibit the messenger RNA from being converted into a protein, which is the translation process. So the interesting thing here is that a single microRNA, it can either target various genes or different microRNA molecules. They can altogether inhibit the expression of a single gene. So this is like increasing the efficiency of a knockdown gene process. You can see that when I'm talking that a single microRNA, it can repress several genes. So this is because several genes, they may differ at a single point with one or two nucleotides, which are known as a single nucleotide polymorphism. So you can see that there is a hairpin loop-like structure in the middle of this microRNA. Due to these SNPs, where this difference lies, they are able to target various genes, and they in turn, increase the efficiency of translational or transcriptional things by inhibiting a single gene. When various different microRNAs, they are targeting a single gene, then the knockdown efficiency, that is further increased. But here it is very, very important to note that when I'm talking that a single microRNA, it can inhibit the expression of various genes. So microRNA characterization and knowing about what are the different genes which are being targeted by a kind of microRNA is very, very important, because if you will go on, modifying microRNA expression, that can downstream inhibit the expression of a number of genes. So characterization of microRNA and its target is the first and foremost very important step when we go on studying microRNA expression or its downstream regulation processes. So about the role of microRNAs in plant growth and development. In plant growth and development, microRNAs, they play a number of functions. For example, they can regulate developmental processes of plants which are related to flowering, which are related to seed development, or meristem identity or meristem identification, or the conversion of a meristem into floral meristem or a vegetative meristem state. Then regulating the expression of regulating the various processes like biotic stress regulation, abiotic stress regulation. When I already told you that most of the studies regarding microRNA expression and its regulation of gene function is being carried out in Arabidopsis, which is a model plant. And let me tell you, it is important to note that a single microRNA family, it can have multiple members. For example, you can see here that microRNA 156, this is regulating flowering. So microRNA 156 family in Arabidopsis, it has almost 7 to 8 members. So that is the diversity of microRNAs, which you can see within the genome. MicroRNA 159, again, it has 7 to 8 members, if I'm not wrong, in Arabidopsis. MicroRNA 156 is a very important microRNA which is regulating the genes which are involved in meristem identity. Then various biotic stress tolerant processes, abiotic stress tolerance processes. So microRNA target genes, the genes which are being targeted by microRNAs, these are involved in regulating various hormonal pathways and other signaling processes and metabolic pathways. So downstream, when a microRNA affects the expression of a certain gene, it cleaves down a mRNA, it is not being translated into a protein, and a certain metabolic process or a certain pathway is being inhibited from taking place within a plant. So that is how this is the process, that how microRNAs, they regulate gene expression. They can target certain genes like transcriptional factors which are regulating hormonal pathways. So in this way, various developmental processes in plant or various processes related to biotic and abiotic stress regulation, they are being affected or they are being regulated by these small tiny molecules which are known as microRNAs. So in apple, microRNAs are post-transcriptional regulators of apple growth and development, fruit quality, hormone signal transduction, and stress. But the studies which are being carried out in woody tree species, they are very, very less. As I already told you, most of the studies, they are being carried out in model plants where they are comparatively easy to study. And in other, woody tree species, the microRNA members, they, in a family, they can be much larger. That is the microRNA family in woody trees that can be much larger as compared to the model plants like Arabidopsis or Nicotiana tabacum. So microRNAs, they are already identified to be the post-transcriptional regulators of apple growth and development in apple, but still the studies, they are in very, very initial stages, and characterization of microRNAs that has not been very efficiently taken up in woody tree species. So you all know about apple. It is a very delicious fruit, and it has a very good antioxidant and other nutritional properties. So winter dormancy, you all know in perennial trees, which is apple is a perennial tree. So winter dormancy in perennial trees, it is an adaptive mechanism, specifically in temperate trees. These temperate trees, they have developed to protect their meristems against low temperature. So in apple trees, cold temperatures, they induce bud dormancy at the end of summer, beginning of fall, and apple buds stay dormant during winter until they are exposed to a period of cold, after which they can resume growth, that is bud break processes, and initiate flowering in response to warmer temperatures in spring. So spring is about to set in, and the bud break events, they have already initiated. They have already started in the perennial trees like apple in this part of the country. It is well known that small RNAs, they modulate temperature responses in many plant species. But however, how small RNAs, they are involved in genetic networks of temperature mediated dormancy control in fruit trees, it still remains largely unclear. So dormancy induction, this occurs when short photoperiods and low temperatures, they set in. That is followed by endodormancy in perennial trees. And during endodormancy, the growth cannot be resumed by favorable environmental conditions. This progresses into ecodormancy. So during ecodormancy, the chilling hours, they have to be satisfied in order for proper bud break, flower set, and fruit set events. So this is a very, very important, and chilling hours, they vary from variety to variety in apple. Some varieties, they require low chilling hours as compared to other varieties of apple which require high chilling hours. And then this eco-dormancy, it is the competence to resume growth after accumulation of enough warmth. And eco-dormancy and then is further followed by bud break, that is breaking of the bud in order to resume growth and either to transition into a vegetative bud or a floral bud. Then that is followed by full bloom in apple. So these are the various stages of bud break, bloom, and fruit set in apple, and these are being recorded at various locations in Thunag. So stage A is a dormant apple bud. You can see here in this picture. B signifies the early green tip. You can also call it as silver tip stage, wherein the bud looks a little furry and silver in color. That is followed by early green tip stage. In figure B, you can see a silver tip stage as well as the early green tip stage. The lower one is early green tip stage and the upper one, that is the silver tip bud. This is followed by a tight cluster of emerging leaves from the bud, and followed by pink bud, and then king bloom, followed by bloom, fruit set, and development of apple fruit finally. So we were specifically interested in studying these two stages, that is A and B, which is a dormant bud and a silver tip bud. Because this is the most significant stage where major molecular decisions after perception of environmental signals are taken, they take place, and the bud has to decide whether it has to progress into dormancy break and it has to develop into a vegetative bud or into a floral bud basically. So these were the two stages of our interest in Malus domestica, that is apple, and we went on mining the microRNAs in apple buds in these two stages, that is the dormant stage, and another one is the silver tip stage, because these are the major stages where important molecular decisions marking the transition of a bud from a dormant stage to a silver stage, they basically take place. So the objective of our study was isolation of quality microRNA from microscopic woody apple bud meristem tissue using laser capture microdissection. This is a very innovative technique as the topic of this faculty development program. It also is based upon innovative research techniques. So this is a very innovative technique which you can use to select a few cells, and then you can go on for your downstream applications in a few cells or tissue within apple or animal tissues. So why studying microRNAs is important in apple bud? First of all, why putting them under a scientific lens is important in apple. One is that microRNA affects the activity of several genes, so they can be one-step target for downregulating the activity of many genes in one step. Second, the activity of microRNAs is expected to have a significant effect on bud development and transition from vegetative to floral bud in apple, which has not been studied till date. So what are the hurdles? What are the hurdles in carrying out this work? It is often very challenging, to study the expression of microRNAs or the expression of genes in difficult to access inner woody tissues of woody tree species, because these are rich in polyphenols and polysaccharides. And when these tissues are rich in polyphenols and polysaccharide, they offer hindrance in isolation of quality pure microRNA or even messenger RNAs from these tissues. So it is difficult to it is also difficult to preserve the inner details, the tissue integrity, and biochemical and transcriptomic integrity due to absence of specific fixation protocols. Most of the protocols, they have been They have been standardized in herbaceous species, while in woody tree species, no specific protocols are available, very much available till date. So we standardized a laser capture microdissection based method for efficient and cost-effective isolation and expression analysis of microRNAs and genes in the meristem tissue of woody apple bud. Why meristem zone? You all must know that this is a very actively dividing zone of cells in plants. So meristem is a very important tissue where major molecular decisions are do take place for breaking of bud dormancy by coordination of extrinsic factors, which are the environmental signals, and the intrinsic factors. So basically meristem is a very important site where these signals are perceived and the bud take decision whether to break or to develop into a vegetative bud or a floral bud. So this is shoot apical meristem. This dome, this signifies the shoot apical meristem, and these are the emerging leaf primordias. This is the section. This is a thin section of a bud. So this is schematic representation of tissue preparation and RNA isolation from apple bud. So we standardize this protocol, and this is a part of my postdoctoral work at National Institute of Plant Genome Research, New Delhi. So this happened, it took almost a week, to fix the tissue and then to proceed for RNA isolation and downstream application. So the bud was excised, and the buds were collected from NBPGR Regional Station, Shimla, and they were harvested and they were immediately transferred to formaldehyde, acetic acid, and acetone, a solution of formaldehyde, acetic acid, and acetone. They were stored in 100% ice cold acetone overnight. So then gradients of 10 to 100% ice cold ethanol, they were prepared in the EPC water, and then followed by transfer to gradients of 10 to 100% terpsputanol. So all these steps, they were, standardized, and various parameters they were set, because apple is a difficult tissue to work with, specifically apple buds, which is about to transition into a silver tip stage, that is bud break stage. So a lot of activity is taking place there, and a lot of metabolites are being synthesized. So isolation of RNA, microRNAs specifically, which are difficult to isolate, it has to be standardized. The tissue has to be fixed, and the tissue integrity and the integrity of biomolecules which are being synthesized within the bud, that has to be standardized. Then we proceeded for making up of blocks. You can see on the right side, making up of blocks and sectioning. This sectioning was carried out in a cryomicrotome, which operates at a very low temperature, because RNA, they are, very damage prone, you can say, molecules, and they are much less stable as compared to DNA. So all this work was carried out under very cold temperature. The sectioning, instead of microtome, instead of a normal microtome, was carried out under a cryomicrotome. Then we proceeded for RNA isolation and quality checking of the isolated RNA. So this is the technique which I was talking about, laser capture microdissection. In laser capture microdissection, what do we do is that we, on the thin sections of a tissue, we project a laser. And when we project the laser, we catapult the tissue, we catapult a group of cells out of the entire tissue, and the entire tissue is it's collected in a microfuge tube, and with those tissues or with those group of cells, you can proceed for your downstream application, which can be transcriptomics, proteomics, metabolomics, whatever you want to see. But this is very interesting that laser capture microdissection, it offers the isolation of few cells out of a tissue. So now with advanced laser capture microdissection, you can also go on checking for your application, whatever you want to study in a single cell, and that has also been done by many labs. So now it has been narrowed down to a single cell. That is, you can see that what is happening, what is being synthesized, what is being metabolized, or what are the transcriptomic or proteomic details of a single cell. We carried out in a group of cells, and our tissue of interest was the meristem of apple. So let me tell you that how this is specifically done. You can see here that this is a thin section of apple bud. We took 10 central sections, which were 10 micrometer in its diameter, you can say, so thickness, sorry. And we took 10 central sections of the meristem of apple, and we catapulted them in a tube. So the red mark, which is, you can see here in the first picture, this is the projection of laser. Now when laser is projected, this part or these few cells, they are separated out of the rest of the tissue, which is being sectioned. And you can see here this gap, this hollow space. From here, this is actually a test. And from here, the meristem of apple that has been excised, that has been apple bud meristem, that basically had been catapulted out of the rest of the tissue. This is ground tissue, which supplies all the nutrition to the meristem for further growth, and these are leaf primordia of apple. So we were, as I already told, interested in two stages. One is dormant bud and another is silver tip stage. So we took 10 central sections from this region of dormant stage apple bud and a silver tip stage apple bud. So in silver tip stage, you can here see that the dome, as compared to here, it is little enlarged or it is little projected. It signifies that some kind of cell division activity, this has already started in the silver tip stage bud, which is the first stage marking dormancy break in apple. Then we proceeded for RNA isolation from dormant and silver tip bud, and we quality checked it on a bioanalyzer. So this has to be standardized, and after several attempts, we were able to isolate a good quality of the RNA and with a RIN value of 7.6, where an RNA concentration of 186 nanograms per microliter. The quantity here is low because it is a few cells from where the RNA is being extracted. So the quantity is 184 nanogram per microliter, which is adequate enough to proceed for downstream application quantity as well as quality. So then we went on for stem loop real-time PCRs of key selected microRNAs from LCM-derived apple bud meristem. We were specifically interested in these two microRNAs, microRNA 156 and microRNA 172. So here you can see the amplification of various microRNAs along with internal control in dormant and silver tip stages of apple meristem in the apple bud. So 156 and 172, these were observed to be present along with microRNA 162, 169, and 166. MicroRNA 156 and 172, why these are important? Because in Arabidopsis, they are already known to control, vegetative to reproductive phase transition in Arabidopsis, which is a model plant. MicroRNA 156 it targets squamosal promoter-like genes, which is a transcription factor which downregulates the activity of several genes downstream. And microRNA 172, it targets AP2. AP2 further downstream regulates the activity of SOC1 and FT, which are the genes which control flowering time and flowering transition in various plants. So microRNA 156 and 172, they were observed to be present and conserved in apple meristem tissue. And we are expecting that these microRNAs, these are controlling the juvenile to reproductive phase transition in apple as well, which is subject to further research, but we were able to see that these microRNAs, they are conserved in apple bud meristem as well. So the function of microRNAs in inflorescence meristem is that you can see here, as plant the phase shift from juvenile to adult stage, down regulation of microRNA 156 increases SPL expression, which in turn promotes the microRNA 172 transcription. MicroRNA 172 is when it increases in expression. MicroRNA 176 triggers the development of inflorescence meristem by reducing mRNA levels of AP2-like gene, as I already told in Arabidopsis, and spatiotemporal functions of microRNA 165 and 66 and their targets, which are at the HDZIP3 genes, which are basically transcription factors, which in turn regulate the expression of several genes. Together with microRNA 164, they restrict the functions of Cooks in specific region of boundary to maintain the inflorescence meristem. So all these microRNAs, they are specifically involved in vegetative to reproductive phase transition in model plants, and we are hoping that this module is conserved in apple as well. And its characterization, it is subject to further characterization as well as research in apple. So coming to the important outcomes of the study, microRNA 156 and 172, they showed conserved expression in apple bud meristem, indicating their potential functional relevance in regulating bud dormancy in apple. And we expect that this kind of LCM-based protocol, specifically as it is, or with some empirical modifications, they would be helpful for RNA isolation and expression analysis of microRNAs or protein coding genes in distinct woody plant tissues, because this has specifically not been carried out to a very large extent in woody plant tissues. Coming to the future prospect, deciphering the role of heat stress regulatory microRNA in temperate horticultural crops as the global temperatures, they are rising. So it is interesting to note that microRNA 156, it also plays important roles in regulating heat stress related responses in various crops. Then deciphering the transcriptome landscape associated with bud development and dormancy break related events in apple. This research could be helpful in these prospects. Lastly, I would like to acknowledge my team at National Institute of Plant Genome Research, my advisor, Professor Ananda K. Sarkar, who is a developmental biologist currently in Jawaharlal Nehru University School of Life Sciences. And this work was being carried out at National Institute of Plant Genome Research in New Delhi as a part of my NPDF program, as a part of my NPDF research fellowship. I also acknowledge the NIPGI New Delhi LCM facility, Central Instrumentation Facility, and the internal grant of National Institute of Plant Genome Research. We also thank Dr. Mohar Singh for guiding with collection of apple buds from ICR NBPGR Regional Station Shimla. And these are all the work which I cited during my presentation. Thank you very much for your patient listening. And I would like to welcome you all to a beautiful abode of Ma Shikari Devi. We are a new college of Dr. Vice Parmar University of Horticulture and Forestry, and we are looking forward for collaboration in research and academics. I welcome you all to this beautiful place. It's snow covered right now, Shikari Devi, but it will open up in April, and you all can, plan to visit this place as well as visit our new college. Thank you very much, all of you. And any kind of questions I would like to take, I would love to take. Thank you so much.
So I have opened the chat box. Anyone having query, they can drop down on the chat box only.
Any questions? I can see somebody said very informative session. Thank you so much. But any queries you have, most welcome. I don't see any.
Ma'am, I think they are full satisfied with their lecture. So
Either they
on behalf
understood
of
every bit of it or they did not understand anything. So I'm
I
really
can see in the chat box like they are thanking you. Very good session, man. There is one query from Gurjeet Kaur, ma'am.
okay. One query, ma'am. Sometime in LCM we face dehydration issues. Okay, dehydration. See, you have to take the water out of the cell. That is for sure, Dr. Gurjeet. The water has to be taken out, and it has to be infiltrated. With your fixatives, which can be formaldehyde, acetic acid, or ethanol. That is what infiltration of the tissue is all about, and you have to take the water content out. But yes, the tissue should not dry up instantly, and it should not die. It should at least preserve its we should be able to preserve its integrity. So these processes, they have to be standardized, and it took almost for us, it took almost more than 3 to 4 months to only standardize the fixation of the tissue and isolate quality RNA. So you have to standardize these things, I would suggest. And I can share, you can go through my paper specifically for woody tissues, because for herbaceous species, these methods have already been well standardized, let me tell you. But for woody species, it is difficult. So that is what I would suggest. And with some empirical modification, I believe that this protocol would also work well with other woody species. So Madhvi, I can't see any other question.
Yes, ma'am. So,
So
ma'am, on the behalf of everyone, I just want to say thank you for giving your time and sharing your insightful lecture with all, ma'am.
thank you. Thank you so much.
Thank you so much, ma'am.
Okay, so I can sign off now?
Yes, yes, ma'am.
Okay, okay. Thank you so much.
Okay, so participants, let's wait for next 5 to 10 minutes for our next guest to join us and having our next session. So I just want to let you know everyone that our next session will begin at 6:30, followed by another session at 6:45. So we are having next two sessions. So I kindly ask you to stay with us and wait patiently until 6:30. So we appreciate for your cooperation and look forward to having you join us for upcoming sessions. Thank you so much.